Hello, welcome back to Godzime. Today we're looking into the regulation of the pyruvate dehydrogenase complex. So in our last video, we discussed how this pyruvate dehydrogenase complex takes part in the conversion of pyruvate into acetyl-CoA and the what makes up this whole complex. So today we'll be looking into how this complex is regulated. All right, so the pyruvate dehydrogenase complex is regulated by two means. We have the end product regulation or inhibition, and we have the covalent modification, which includes phosphorylation and dysphosphorylation. All right, so the pyruvate dehydrogenase complex is always inactive in the phosphorylated form. So once it's phosphorylated, it is inactive, and in the dysphosphorylated form, it is active. So from here now, we can see the from this diagram here, we see this is the pyruvate dehydrogenase complex, and it is regulated by two main enzymes. We have the pyruvate dehydrogenase phosphatase, okay, that removes this phosphate. This actually dephosphorylates it, and we have what here, the pyruvate dehydrogenase kinase that phosphorylates it. All right. So now, just like I said earlier, I said it is regulated by two means: end product inhibition and also by covalent modification. All right, so here we have the pyruvate dehydrogenase kinase. All right, now what kinase actually does, from our last video on that glycolysis, is say kinase what phosphorylates. All right, we say kinase phosphorylate, which means it adds up phosphate groups on what, on this complex, on this pyruvate dehydrogenase complex. All right, so once it adds phosphates to this pyruvate dehydrogenase complex, this complex becomes inactive, which means the process of aerobic glucose oxidation cannot be carried out again because this complex is already inactive. All right, so let's look at what stimulates this pyruvate dehydrogenase kinase to phosphorylate this complex. Remember, we said end product inhibition. So from the last video we talked about, we talked about an end product. The end product of this were acetyl-CoA, ATP, and we also listed NADH, which was given off, that went through the electron transport chain. All right, so these are the end products. All right, so once these end products are merged in the body, which means they will come what, end product inhibition, they will come what, and they will stimulate this pyruvate dehydrogenase kinase, and they cause this pyruvate dehydrogenase kinase to phosphorylate this complex, to stop this complex from actually carrying out the aerobic glucose oxidation. All right, so these are the end product, and they stimulate this enzyme. All right, so what this enzyme does, as I said earlier, it phosphorylates. All right, it phosphorylates, which means it adds phosphate groups on this enzyme. All right, it phosphorylates this enzyme. So phosphorylating this enzyme, it inhibits it. Phosphorylating this enzyme, it inhibits it. All right, it inhibits this enzyme. All right, so phosphorylated form of pyruvate dehydrogenase complex is actually inactive. So this compound acetyl-CoA, okay, stimulates this enzyme, because when there are lots of acetyl-CoA in the body, all right, which means lots of aerobic glucose oxidation are going on in the body. I know this acetyl-CoA actually has to be given off into the Krebs cycle to yield energy. All right, which means when there are a lot of acetyl-CoA, we have to stop the aerobic glucose oxidation. So acetyl-CoA inhibits stimulates this enzyme pyruvate dehydrogenase kinase. All right, and this stimulated kinase phosphorylates this enzyme, and it inhibits this complex. All right, so that's how acetyl-CoA inhibits this complex. All right, so the next one is what? ATP. All right, remember in the last part of this in the last video we talked about the giving off of ATP, or because the NADH and the H ions actually have to go through the electron transport chain to yield ATP, and also the acetyl-CoA that was given also into the Krebs cycle we yield ATP too. All right, so when there are a lot of ATP in the body, all right, so this ATP will come and to stimulate this pyruvate dehydrogenase kinase. So once this kinase is stimulated, it what phosphorylates this complex and what thereby what inactivating it, which means it inhibits this complex from carrying out what this aerobic glucose oxidation, and the level of glucose what will be the level of ATP will be maintained. All right, so NADHs, all right, so NADHs also stimulates NADH, all right, because NADH has to go what into the electron transport chain also to use ATP, all right. So NADHs, when a lot of NADHs, what it stimulates this enzyme, so causing this enzyme to what to phosphorylate this complex and inhibits it. Okay, so now let's talk about what inhibits this kinase or pyruvate dehydrogenase kinase, because this end products we've measured now are actually stimulating it to phosphorylate this complex. So what are the things that inhibit this pyruvate dehydrogenase kinase that will make it not to phosphorylate this complex? All right, the thing that actually inhibits this complex are pyruvate, because when there are lots of pyruvate in the body, which means we have to carry out aerobic glucose oxidation, which means this process has to go through this enzyme has to carry out this aerobic glucose oxidation. All right, which means pyruvate comes here and stimulates. All right, pyruvate comes here and inhibits. All right, pyruvate comes here and inhibits this complex, this enzyme here. So pyruvate comes here, and inhibits this pyruvate dehydrogenase kinase, preventing it from phosphorylating the pyruvate dehydrogenase complex. All right, also NAD positive, all right, inhibits this complex. NAD positive inhibits this complex. All right, and also this complex also inhibited by CoA, So these are the inhibitors and the stimulators of this pyruvate dehydrogenase kinase. So acetyl-CoA, ATP, NADHs, okay, they stimulate this pyruvate dehydrogenase kinase to phosphorylate this pyruvate dehydrogenase complex, thereby deactivating it, all right, because they inhibit this compound. All right, so pyruvate NADH positive and CoA comes and they inhibit when there are lots of pyruvate in the human body. All right, it inhibits this pyruvate dehydrogenase kinase, preventing it from phosphorylating this kinase. All right, so preventing of phosphorylating this pyruvate dehydrogenase complex can actually carry out the process of aerobic glucose oxidation. All right, so this is how pyruvate dehydrogenase kinase is actually regulated. All right, so now let's talk about the pyruvate dehydrogenase phosphatase. Seeing this phosphatase means they take off phosphate group, they remove phosphates from this complex. All right, remember the phosphorylated form of this complex is inactive and the dephosphorylated form is always active. All right, so this is the covalent modification, phosphorylation and dephosphorylation. All right, so let's look at now what actually stimulates this enzyme to dephosphorylate this complex, so that this complex can take part in the aerobic glucose oxidation. All right, so now here we have our pyruvate. All right, so when there are a lot of pyruvate in the body, all right, pyruvate stimulates this enzyme. All right, it stimulates this enzyme, and what this enzyme does is it dephosphorylates. Okay, it dephosphorylates it. All right, it dephosphorylates. I mean, it takes off the phosphate groups. Okay, it takes off these phosphates, it removes them. Okay, it moves the phosphate groups. All right, so pyruvate is stimulate this enzyme. All right, also we have the NAD positive. All right, the NAD positive that stimulates this pyruvate dehydrogenase phosphatase to remove the phosphate group, so that this complex becomes very, very active. Okay, we also have the magnesium ion, we also have the calcium ion. Remember, for skeletal muscles to actually move, there should be due to an increase in the action potentials, there's an influx of calcium ions from the sarcoplasmic reticulum. All right, because the sarcoplasmic reticulum have ryanodine channels, which are calcium channels. All right, so an influx of these calcium ions cause the skeletal muscles to actually move. And this skeletal muscle, for them to move, they require what? ATP too. All right. Remember, ATP in the body is broken down to ADP and also AMP. All right. So in a reduction in ATP, which means there's an increase in ADP and AMP. All right. So when there's an increase in AMP and ADP, which means there's a reduction in ATP. All right. This is actually what stimulates this pyruvate dehydrogenase phosphatase to remove the phosphate group, so that this pyruvate dehydrogenase complex can actually what carry out the process of glucose oxidation, all right, to give ATP in the body, so that the body have a lot of ATP for what, for skeletal muscles to actually carry out their work and for us to actually use in our body. So AMP and ADP are actually what stimulators of pyruvate dehydrogenase phosphatase. All right, so these are the stimulators of this enzyme that dephosphorylates this complex, and these here are the stimulators of this enzyme that phosphorylates it, and these are the inhibitors of the enzyme that prevents the phosphorylation of this complex. So this is how the pyruvate dehydrogenase complex is actually regulated in the human body. So now let's move on into the clinical correlation, clinical relevance of the pyruvate dehydrogenase complex and everything we talked about in aerobic glucose oxidation. All right, so remember we talked about thymine pyrophosphate. Yeah, we talked about thymine pyrophosphate, TPP. All right, thymine pyrophosphate, we said what? It is derived from what? From vitamin B1. Vitamin B1, which is what? Thymine. Thymine. So when there is a deficiency of thymine pyrophosphate, which means there's a deficiency of what? Thymine. All right, this can cause what? Deficiency. I'm coming. Okay, deficiency of TPP. Okay, the main constituent of thymine pyrophosphate is thymine. So when there's a deficiency of thymine, which means the process of aerobic glucose oxidation cannot occur because there's a deficiency of thymine pyrophosphate. All right, so the pyruvate will be accumulated in the human body. The pyruvate will not be converted to what? Acetyl-CoA. All right, so there's an accumulation of pyruvate in the body. So there's deficiency of TPP. So there's an accumulation of pyruvate. All right, so when pyruvate is accumulated in the body, all right, so pyruvate will be converted to lactate. Will be converted to lactate, and lot of conversion of pyruvate to lactate will lead to what? Lactic acidosis. Lactic acidosis. All right, will lead to lactic acidosis. All right, also when there's a deficiency of thiamine, all right, it causes a medical condition known as beriberi. All right, so beriberi has the symptoms such as what? Tiredness, nerve disturbance. We also have paralysis. All right, because there's a deficiency of thiamine, we also have paralysis. We also have dyspepsia, which is pain in the stomach. Dyspepsia. Okay, dyspepsia. All right, so these are the symptoms of what? Beriberi. All right, so deficiency of thiamine may also cause a medical condition, a neurological condition known as Wernicke's encephalopathy. All right, may cause Wernicke's encephalopathy. All right, so this is when there's deficiency of thiamine, and this Wernicke's encephalopathy is characterized by what? 3 clinical triads, such as what? Confusion, ophthalmoparesis, Ophthalmoparesis, which is what? The paralysis of the extraocular muscles, okay? Paresis. And this is also what we also accompany with what? Nystagmus, and we have lastly anthasia, all right? Anthasia, lack of coordination of the muscles, all right? Confusion, ophthalmoparesis, which is paralysis of the extraocular muscles, all right? This may cause nystagmus, and lastly, we have anthasia, lack of coordination of the muscles, all right? So these are the conditions that we develop due to a deficiency of thiamine, all right? So if in Wernicke's encephalopathy, what we have to do is we have to what? Give what? Thiamine therapy, okay? We have to give thiamine therapy to the patient, all right? Now, if this condition, Wernicke's encephalopathy, is not treated, we may develop what is called the Korsakoff's syndrome, all right? Korsakoff syndrome, and this is not reversible anymore, all right? Because now this affects what? Some parts of the brain, such as the brainstem, the cerebrum, the thalamus, and the hypothalamus, all right? So now, in also patients with inherited deficiency of the pyruvate dehydrogenase complex, lactic acidosis is actually what is actually seen in these patients also. All right. So the deficiency of thiamine may lead to what? Lactic acidosis. Okay, may lead to beriberi, which has these symptoms, and also will lead to what? To Wernicke's encephalopathy. And if Wernicke's encephalopathy is not treated, it may develop what? Korsakoff syndrome. So sometimes we call it Wernicke's Korsakoff syndrome, because untreated Wernicke's encephalopathy leads to Korsakoff's syndrome. So this is the clinical relevance of the pyruvate dehydrogenase complex and what really happens in the regulation of pyruvate dehydrogenase complex. So if you enjoyed this video, please give us a like and subscribe to this YouTube channel for more videos, and don't forget to leave a comment in the comment section below.